Hi everyone, we are VisiCELL, GPS for cell therapy. My name is Mya Thu, founder and CEO of VisiCELL. We provide non-invasive imaging-based cell tracking solutions to our target customers in the multi-billion-dollar cell therapy industry to assist with safety and efficacy assessment for product development acceleration and improve patient outcome. I'm a biomedical engineer by training and have masters in medical device and diagnostic engineering. And I have more than 15 years of experience in this space, having trained at prestigious institutions such as NIH, City of Hope, and industry startups, including MI Bio Research, which was acquired by a large CRO, Covance. There is no better way to illustrate the potential of cell therapy than Emily. Emily is living cancer-free for 8 years after receiving CAR T-cell therapy developed by Novartis. However, translating the success of cell therapy from blood cancers to solid tumors have been extremely challenging, to say the least. The cell therapy developers face major roadblocks imposed by solid tumor that negatively impact the cell therapy behavior and performance in many ways in the subject. To name a few, tumor microenvironment, heterogeneity, and shared antigen target with normal healthy tissues all contribute to unpredictable cell therapy, biodistribution, homing, and infiltration, and on target of tumor toxicity, and challenges with optimal dosing while balancing benefits and risk. There are direct clinical consequences associated with these major roadblocks by solid tumor. For example, unsuccessful therapeutic outcomes, while there is astounding therapeutic efficacy in the dish. The problem is, currently, we do not know whether or how these cell therapy products are hitting their marks or not in the patient as intended. It is absolutely critical to track the cell therapy products to understand their tumor engagement to make a determination whether the cell therapy product has a favorable impact or not with proper tumor engagement for critical, timely decision-making for product development and treatment strategy. Without cell tracking solution to make such determination, the current means to measure the outcome, for example, the attributes from cancer spread or growth, take too long to show. VisiCELL has the solution that tracks the cells in real time to monitor their solid tumor engagement in subjects, and is simple and easy to use. Mix our patent-pending biodegradable nanoparticles with the cells, whereas the cells take up the particles and inject those labeled therapeutic cells to the subjects and observe using standard MRI available in most clinics. The potential clinical applications of real-time cell location information and their tumor engagement are treatment efficacy associated with proper tumor engagement, dosing regimen and strategy, for example, repeated dosing versus larger dose for effective tumor infiltration, or identifying patients who will benefit from specific cell therapy product. Using our core technology, Su et al. At Northwestern has elegantly illustrated how early targeting of immune cells to the tumor site can inform us the future therapeutic efficacy or response. Natural killer cells, NK cells, were injected to the rats harboring tumors via hepatic artery. By MRI, we can see NK cells home and infiltrated at the tumor site within 48 hours after injection. The number of cells infiltrated at the tumor site, indicated by the changes in MR signal intensity, directly correlated with the reduction in tumor volume. This demonstrates the use of imaging as an early biomarker for treatment response as a companion therapeutic. The prevailing standard of care at this time is grind and find PCR and painful and invasive biopsy. While they are easily accessible, it only provides one single time point, one single location, which doesn't address the challenges of the solid tumor. PET imaging, while it provides whole body outcome measures, it uses radioisotopes and radiation to take an image and is highly undesirable for the cancer patients. VisiCELL positions itself at the right corner where we use standard MRI available in most clinics for easy accessibility and easy integration into current existing tumor response monitoring while providing whole body outcome measures. Our the competing technology is 19F modified MRI, which requires specialized equipment and hardware that's not available in most clinics. For example, less than 1% of 12,000 MRI machines in the United States has such capability, and even then it requires specialized staff training, and the hardware is not for every body part. VisiCELL provides real-time cell location information for early and ongoing outcome measure at 20 times cheaper and 5 times faster while reducing the patient's burden and integrating it easily into existing workflow. VisiCELL's underlying core technology uses iron-based biodegradable nanoparticles that are internalized by the label cells as shown in TEM of T cells, MSCs, and NSCs here. We have also demonstrated in Nature Medicine paper that there is no significant toxicity detected in these label cells by apoptotic assay, metabolic assay, and redox assays. There is also no effect on functionality or differentiation capability of these label cells. Tracking of cell therapy product by standard MRI and using our underlying core technology extend beyond immune cell therapy product. What is demonstrated here is genetically engineered neural stem cells that are used in glioblastoma model of mice. Here, MRI and our with nanoparticles show that homing of label NSCs via different route of administration intratumorally on the left side, left panel, and intravenously on the right side, detected at the brain tumor site and up to 12 weeks. The technology has also been demonstrated and validated by the third-party laboratory in regenerative medicine application. What we are looking at here is using standard MRI technology and these iron-sensitive, MRI-sensitive nanoparticles, MSCs can be tracked in Alzheimer's models for up to 14 days. We have a great team with more than 40 years of directly related experience. Dr. Johnny Akers, Director of R&D, has both nanoparticle characterization and development and cell therapy in cancer model. Dr. Amit Jandali, co-founder and practicing radiologist, provides us customer insights to help facilitate customer integration into existing clinical workflow of our customer and facilitated adoption. Myself as a founder is the inventor of core technology as well as the follow-up patent pending technology. We have accomplished advisory board of academic and industry oncologists and regenerative medicine specialists, serial entrepreneur, and radiologists that are helping us and guiding us our strategy on how we can help solve this unmet need in the field. The benefits of real-time cell location information span from preclinical development to clinic. For example, in preclinical stage, the technology can help select lead candidate in vivo, which we all know that in vivo performance of a product is different from in vitro, that it is essential part of the study. In IND enabling study, the technology can help help with FDA required biodistribution and also assist in analysis of tox efficacy study. This real-time cell location information has tremendous value in early phase clinical trial and can guide analysis of efficacy data. For example, if 3 patients out of 10 patients respond, why are they responding? Is it associated with proper tumor target engagement or the retention of the cell therapy product at the target site? In clinical setting, VisiCELL GPS tracking system for targeted cell therapy can confirm the location of the cell therapy product to the target site, which allows the clinicians to predict and analyze the effectiveness of the therapy much sooner than the current clinical means, and can act as a companion therapeutics. All of these benefits help with accelerated development time to market and rapid clinical decision making and ultimately improve patient outcome. We have fully developed different products that are tailored toward different stem cell type or immune cell types and their need, and have validated in vitro assays and in vivo model. We are looking for partnership opportunity at this time. We are open to collaboration for evidence-based use cases and co-development partnerships for new product creation that will help solve your current unmet need while mutually beneficial for VisiCELL. We are also looking for strategic partnership with biopharma cell therapy companies to establish our technology as imaging-based companion therapeutics that can be used to predict therapeutic outcome of cell therapy product in cancer. Together, let's put GPS cell tracker in every cell therapy. And I'm looking forward to speaking with you in one-on-one partnership meeting. Thank you.